So our first actual section of material today is just a general introduction to development and some of the major concepts of development. As usual, we'll start with the definition. Development just refers to all of the changes that occur during an entire life cycle in an organism. So we're not talking just about embryonic development. Development continues after birth or hatching or whatever process you're dealing with. So in humans, for example, you have embryonic development, which lasts about 40 weeks, but human development doesn't stop at birth. Obviously, newborns continue to grow and develop through their childhood, adolescence, and into adulthood. So development refers to all of those processes, not just embryonic development. It's really difficult to study embryonic development in people. There are logistical and also ethical questions associated with that. So what we can do instead is use model organisms, and there are a number of species that are really easy to study when it comes to embryonic development. You can see some pictures of these here. Now, you don't have to know what these organisms are, but we're going to actually use different parts of these specific development pathways as examples, or at least some of them, 3 out of the 4. So this right here, this is a worm called C. elegans. I've mentioned C. elegans before. It's a nematode, it's a roundworm, and it's a protostome, so they have determinate cleavage. So we've actually mapped out that entire developmental pathway from zygote to the adult, and the adults are tiny. They're only about 1 millimeter long, so they only have about 1,000 cells, a little over 1,000. So we've actually mapped out that entire pathway. Other organisms that are really easy to use are sea urchins, frogs, and birds, specifically chickens. And as we go through these 2 lectures on development, we're actually going to see different parts of the processes illustrated in these 3 model organisms. Remember, the point of a model organism is something that illustrates a process in a different organism, but is also easier to study than that target organism. So humans, again, are tricky to study, but sea urchins, frogs, C. elegans, and chickens are relatively easy to study, and we could use them to inform our knowledge of other species. So one other big idea before we get into the specifics is that development is an ongoing process. As I said a minute ago, it doesn't just stop at the end of embryonic development. And we can use frogs as an example here. So we have our gametes, our sperm and egg. They're going to fuse into a zygote. That part of embryonic development is called fertilization. So here in this box, we have embryonic development. And then we have cleavage, gastrulation, organogenesis, followed by, in this case, hatching. And then we have development throughout life, from larva to adult. Now we, in these 2 lectures, are only going to talk about embryonic development. So not this time, but next time, the last thing we're going to talk about is human birth, and then we're going to end it right there as far as development goes, and we're going to jump right into other systems in the body as they work in adults. So we're just going to kind of skip that childhood and adolescence part of development. We're only going to talk about embryonic development, and then that will lead into the adult physiology. So that's our introductory section. The next thing we're going to talk about is fertilization.